I think this is important because this is the main test that is done in many centers, in all centers. In fact, for blood grouping, this is the main basic test. So, and this comes in all questions also regarding the use of antiglobulin reagents, how the quality control of these reagents, and also the how we use the polyspecific, the monospecific reagents, and their quality control, and as sort of questions also come, and also regarding the anti how we test for them on the tube testing, the gel testing, and the solid phase. So this was something I, if we like go through it just on our own, it kind of gets some really sometimes you can just like get stuck in some point. So I thought it would be better if you all like pulled in together and were able to do it, since it's sort of a very technical also in some uh, ways. So we'll just go through, I would say, some sort of speed reading of this thing, of this book. So now if we go with the history of the antiglobulin test, I think now they are just basically I'll just go through some important points and we can mark them along. Now, the antiglobulin test is also the Coombs test, of course, which is the based on the principle of the anti-human globulin obtained from immunized non-human species which bind to the human globulins, such as immunoglobulin uh, or complement either free in serum or attached to a antigen or so this is actually even this could be like an anti-globulin test, which will have the IgG component. It could be IgG or IgM, and this could bind to the human globulins such as IgG or complement, either free in serum or attached to the antigens on the red blood cells. So when they're attached to the antigens, we could use it as a direct Coombs test, and if they are free in serum, we could use that this Coombs reagent could be used as a indirect antiglobulin test. Now, the AHG test is an essential testing methodology for the transfusion medicine, and there are two major types. Basically, it could either test the reagent itself is IgG, is based on it binds to the human immunoglobulins or the complements. The reagent itself, I think, it's an anti-IgG. So it's an anti-IgG. It contains the anti-IgG, which and it can bind to basically immunoglobulin G and the complement also. And once it binds to this binding to complement would be how it would be used to detect the IgM activation. So in a way, this anti-IgG, since it can bind both to IgG and the complement system, it can detect both these antibodies. So because of their large pentameric structure, the IgM antibodies bind to corresponding antigen and directly agglutinate the RBCs suspended in saline. Some IgG antibodies are termed non-agglutinating or incomplete antibodies because their single monomer structure is too small to directly agglutinate or sensitized RBCs. Of course, we know that this is a pentameric structure, so this can directly agglutinate, and and the agglutination can be even seen with the naked eye. But some these IgG antibodies, they are non-agglutinating because they are small and their agglutination cannot be directly seen with the naked eye, and in fact they may not form such agglutinates that can be picked up. So adding and so we need to add certain reagents, and this AHG reagent, this containing the anti-IgG. The AHG reagent containing the anti-IgG then allows for hemagglutination of these sensitized cells. Now, an antibody is used to observe the formation, an anti-antibody is used to observe the formation of these antigen-antibody complex. So we have basically the antigen which binds to an antibody. This antibody can be free in serum or it can be attached to the RBC. So this antibody is actually the anti-antibody that we are introducing, and the antigen can be either on the red blood cell surface, or it can be bound to the or it can be free in the serum. This antigen can be either on the red cell surface or it can be free in the serum, and and the antigen may in itself be an immunoglobulin. So it could itself be an immunoglobulin, either the IgM or the IgG type, And the antigen antibody complex would then be detected by the anti IgG. So some blood group antibodies have the ability to bind complement to the RBC membrane, and in such cases, an anti-complement component can be added to the AHG reagent, so rendering it as polyspecific. So anti-IgG and anti-complement, they can both be detected in this way. So anti-globulin tests detect the immunoglobulin or the complement sensitized RBCs. So this was like basically about the Coombs reagent, that it is an it is an anti-antibody in the way that it is an anti-IgG basically, that will detect the IgG, which may be bound to the red blood cells in the direct Coombs test, or it may be in the serum, if free in the serum, because of in which case it might be detected as the indirect in immunoglobulin. And this not only binds to the immunoglobulins, but it can also bind to complement factors. So this anti-complement component, this can be added to the AG reagent, and in this case it would be polyspecific, that it would have not only the anti-IgG component but the anti-complement component also. So historically, this was used because to detect the antibodies in serum and those on present in the red blood cell surface. So and this its first use was actually in HTFN measurements, because the test was originally even called the Coombs D8 test, after the Coombs this Coombs person who used this to detect the in vivo sensitization of the RBCs of neonates suffering from hemolytic disease of the fetus and newborn. The test was originally called the Coombs test, but later was called the direct antiglobulin test. So these are both synonymous names, the Coombs test and the direct antiglobulin test. Although Coombs and associates were the instrumental in introducing the antiglobulin test to the blood group serology, the principle of the test had in fact been described. Okay, so this was sort of this is the basic history, that its first use was in HTFN. Now the Coombs test involved the injection of human serum into rabbit to produce the anti-human serum. So, so the reagent historically it was derived from rabbits who had been injected with human serum. So in a way they had this anti-human antibodies. So after absorption to remove the heterospecific antibodies, and after dilution studies, so basically heterospecific antibodies, that is those relevant only to the rabbit itself, those non-specific heterospecific antibodies were absorbed, and then after dilution studies, and to avoid the prozone interference, the AHG serum. Now this prozone is actually in this prozone effect is there in most cases. There is an anti this after so you would dilute it so as to dilute out the antibodies, because excess of antibodies can also lead to a prozone phenomenon, sort of a false agglutination. So the AHG serum still retains sufficient antibody activity to permit cross-linking of adjacent RBCs sensitized with IgG antibodies. Now, the cross-linking of these sensitized RBCs by IgG produced the heme agglutination, indicating that RBCs had been sensitized by an antibody. Okay, so if we add now the AHG reagent, which has the human specific, it is an anti-human globulin, so that's why it was, because it was first derived from rabbit sera. So the non-heterospecific antibodies were absorbed and then were absorbed out, and then remaining antibodies were diluted to avoid the prozone interference. And then these antibodies, they still retain the sufficient antibody activity to permit cross-linking of adjacent RBCs sensitized with the IgG antibodies. So once this reagent was added to antibodies, to RBCs that had been sensitized and they had the antibody on the surface, so they could react with with those cells and cause agglutination. So early AHG reagents were prepared using a crude globulin fraction as the immunogen. And Then Desi also presented the first evidence that there will be another antibody activity present. And he observed that direct reaction patterns were obtained when dilutions of AHG were used to test cells sensitized with warm IgG. So dilutions of AGG, if they were used, different reaction patterns were obtained when dilutions of AGG were used to test cells sensitized with warm IgG as compared with cold IgG. So and the activity of AHG to cells sensitized with warm antibodies resulted from the anti-gamma globulin activity, whereas non-gamma globulin activity was responsible for the activity of cells sensitized by cold antibodies. Okay, so if he observed that different reaction patterns were obtained when dilutions of the AHG were used. So if you use the dilutions of AHG, so AHG has IgG component, and if you use dilutions of this, then different reaction patterns could be seen. For example, if we test cells which have been sensitized with the IgG component, then of course the IgG, the anti-IgG would react in a different way. But if they were, if the cells were coated coated with cold antibodies, IgM, which are reactive at less than 22 degrees centigrade, then a different pattern would be noted. And this would be, of course, because the IgM antibodies, they also activate the complement. And so this non-gamma globulin component was shown to be beta globulin and had specificity for complement. So this is where the complement system also comes in. So the AHG is actually anti-IgG, but it has an anti-complement effect also. And because of that, it would still react with the RBCs that then may not have the IgG, but still have the complement component, that is the beta globulin part. So they are not gamma globulins, but they are beta globulins. And later studies revealed that the complement activity was a result of the C3 and C4 complement factors. So the C3 and C4, they caused the activation of these activated complement beta globulin parts on the RBC surface, and that could also be detected by the anti-IgG component. So the antiglobulin test can be used to detect RBC sensitized with IgG allo, and so both allo, so not only the IgG but also the complement C3 and C4 beta globulin parts can be picked up, and then also all allo and the auto antibodies can also be then picked up. Now sensitization can occur either in vivo or in vitro. So not only in vivo also, but so basically we have this anti-globulin this AHG, this anti-globulin reagent or the anti-IgG reagent. It can react both with these gamma globulins and then with the beta globulins, which are the complement factors with C3 and C4. So it can react with IgG also and with complement also. And then it can detect allo antibodies also and auto antibodies also. And then it can react with and then it can and then it can react this sensitization can occur in vivo or in vitro. So even if they are bound to the RBCs, even they then can be detected, or if they are lying in the serum, they then can even be detected. So so in all these, so because of the wide basically specificity of these antiglobulins, they have a very wide range of use that we can use them for. Because of this, the tests that can be done, they are actually one stage. When it's a one-stage procedure, this is the direct antiglobulin test, and in a two-stage procedure, we can even indirectly detect the antibodies lying in the serum. So several AHG reagents have been defined by the FDA, and so anti-human globulin. So it can be both a polyspecific or monospecific. So now the polyspecific AHG is this contains the human IgG. This contains the antibodies against the human IgG and the C3d component of the human complement. So and other anti-complement antibodies such as anti-C3b may also be present, and therefore its use can facilitate the agglutination when RBCs have been sensitized with IgG or C3d or both. So so the polyspecific, they would these bind to both the IgG component and the C3D component. They can bind to both, or they can bind to either one of them, and they are usually derived. Since historically they have been derived from rabbit serum, so most of them are still derived from rabbit. Although murine monoclonal blends can also be there, but the monospecific one would be then IgG, and they would be either anti-IgG specific, or with the depending upon the clone which is used, it could be a rabbit polyclonal or a gamma clone AHG, and these are the murine derived, so they could be rabbit or murine derived also, and or they could be anti-complement or anti-C3D. So they could, if they are monospecific, they could be specific only against IgG, or if they are, they could be specific only against the C3D, and then these are those against the complement. They are actually the main component of this reagent is the murine monoclonal antibody to C3D, and this anti-C3D will then cause the agglutination of cells coated with human C3D or C3D complement components. So so this is how they have actually divided. It's polyspecific, would have both of them in the same reagent, and it could be either rabbit derived or murine derived. And the monospecific would have either the IgG component or the C3D component, and this could also be rabbit derived or murine monoclonal antibodies from hybridoma cell lines. So they could be like specific for one component only. So the polyspecific have IgG or C3D, and the monospecific AG, they contain only one antibody with specificity either for the anti-IgG or C3D or C3b. So it could be either C3D or C3b. And licensed monospecific AIG reagents in common use are the anti-IgG, anti-C3b, or C3D. C3D is actually this is the complement component which is derived from the further breakdown of C3D by the macrophages. So now the reagents they labeled anti-IgG contain no anti-complement activity. So those which are anti-IgG are they have no anti-complement activity, and these contain antibody specific for the FC fragment of the gamma heavy chain of the IgG. So since they are directed against the IgG, they are directed. These are the gamma heavy chains. The IgG will have the gamma heavy chains, and they will be directed against the FC fragment of the gamma heavy chains. If not labeled gamma heavy chain specific, anti-immunoglobulin can contain a light chain specificity as well, and may therefore react with cells sensitized with IgM, IgA, and IgG. So this is how even the monospecific IgG, although they are specific for IgG, they still may have sort of a reaction with weak IgM, IgA antibodies also, because of the light chain specificity. So if they are not these anti-IgG, if they are if they have antibodies specific for the Fc fragment of the gamma heavy chains of the IgG, then they are specific for only the IgG. But if they have specificity against the light chain specificity, then they can cross react with cells sensitized with the IgM or IgA. So if we have like a monospecific AIG, then this could be either the anti-IgG, but even that could be directed both against the IgM or the IgA or the IgG, even if it is not specific for the gamma heavy chain. So and the anti-complement, this can also then have mixed reactivity against the C3b or the C3d reagents. and this can contain no activity against this would have no activity against human immunoglobulin. So anti-immunoglobulin would have no complement activity, but the immunoglobulin fraction could be either IgM or IgA. And the C3 and the anti complement reagents, they would have no human globulin and specificity, but they could be a blend of monoclonal anti C3b or C3d. So another preparation of these anti-human globulin um, is that this involves basically the injection of human sera into the laboratory animals such as rabbits, and the human globulin then behaves as foreign antigens, and then the rabbit's immune system is triggered and an antibody to the human globulin is produced, of course, and then this is then injected. This is injected into um, uh, this can be then used. This anti-human globulin, this anti-human globulin from this rabbit can then be purified by absorbing out the heterospecific rabbit antibodies, and then diluting the resultant antibody to absorb to prevent the Prozon phenomena. And human complement the human IgG injected into a rabbit results in anti-IgG production, whereas human complement components injected into rabbit result in anti-complement fraction. So they can be derived in different ways. If only human globulins are injected, we'll get anti-human globulins. If human complements are injected, we would get anti-complement component. And then these and polyclonal antibodies would be a mixture of all these antibodies, whereas monoclonal antibodies are basically derived from a clone of plasma cells, and they recognize a single epitope, and these are derived using hybridoma technology. So the polyspecific AHG was then can be used made using polyspecific polyclonal or monoclonal antibodies, and the two types of antibody production processes are very different from one another, yielding two very different advantages and disadvantages in their usage. So the polyclonal, in fact, this can also be the polyclonal and the monoclonal AHG. So the monoclonal is the one that is derived from the immunization of laboratory animals and with purified human globulin. So, and the polyclonal AHG is derived from it is derived from laboratory animals in which by immunizing one colony of rabbits with human immunoglobulin antigen, and another colony with human C3 antigen. So, and there will be a heterogeneity of the IgG molecules. So that's why they're called polyclonal, because they actually have these uh, monoclonal is actually injected into in monoclonal they actually inject these purified human globulins, um, which are then after a suitable immune response, the mouse uh, uh, when they start secreting those antibody specificities, those lymphocytes are then fused with myeloma cells. Okay, so monoclonal is always going to be by hybridoma technology, whereas the lymphocytes which are obtained from the spleen, those lymphocytes which have started producing antibodies against those purified human globulins, they are then these lymphocytes which are obtained from spleens, which are producing antibodies against these purified human globulins. So those lymphocytes are then fused with myeloma cells, and this hybrid would then be used to produce antibodies of required specificity and affinity. So the monoclonal is always going to be made by hybridoma technologies, and whereas the polyspecific AHG is going to be made by immunizing a colony of rabbits with so they would be using a colony of rabbits with the one with the IgG component, other with C3 component, and the antibodies produced would then be pooled together to make this polyclonal reagent.